Hey, welcome to video 18, unit 1.3, how cells make protein. So, we already learned about this idea, the central dogma, which basically tells us how genetic information flows through the cell. So we know that it starts out as DNA, and then through a process called transcription, which happens in the nucleus, a gene is copied as RNA. RNA then leaves the nucleus, and through a process called translation, gets used to make a protein. And that happens in the cytoplasm, which is basically anything outside of the nucleus that's still inside the cell. So this is our central dogma. DNA, RNA, protein. These processes are called transcription, translation. They happen either in the nucleus or in the cytoplasm. So the thing about proteins is that they're made for a huge number of different reasons. Some proteins are used directly by the cell, and when that happens, the process is very simple. You have transcription take place in the nucleus, the piece of RNA finds a ribosome in the cytoplasm, the protein gets made, and it goes to work. But a lot of proteins are actually exported through the cell membrane. They're pushed out of the cell. In multicellular organisms, this is really important. So these are proteins that, they might be hormones, they might be necessary for the communication between cells, or, they could be types of enzymes, like the cells that make up your salivary glands producing an enzyme called amylase that helps to break down starch. When those things are made, the process is a little bit more complicated. So you still have transcription happen in the nucleus, but the little piece of RNA doesn't go to a ribosome in the cytoplasm. It goes to a ribosome in the rough ER. And when translation happens, that's not the end of the story. The protein then gets further processed by the ER and by the Golgi apparatus. So here's the cell, and I've just highlighted a few things inside of it, like the nucleus and the rough ER, and then the Golgi apparatus. And this is nothing like what they would look like in an actual cell, because they're not purple and red and blue. But it helps to teach people using bright colors. And also, the ribosomes are not star-shaped. So, bear with me. So anyway, so in the nucleus, you've got the DNA, the permanent copy of all the working genes that the cell has. And so in order to make a protein, the instructions for that protein, stored as DNA, have to be converted into RNA. So that's the first step in this process. Transcription, right? Taking DNA and making a copy of a gene in RNA form. So that happens in the nucleus. And then that little RNA can find its way to a hole in the nuclear envelope into a ribosome in the rough ER. And once it's there, translation happens. And at the end of that, you have what's called a polypeptide. It's not a protein yet because it's not a functional unit, but it's a chain of amino acids. And it still has to do, the cell still has to do some work to it before it's finished. So for example, after the polypeptide is made by the ribosome, it moves through the rough ER, and signaling sugars get attached to it. And those signaling sugars identify the parts of the polypeptide that still need further work by other parts of the cell. Eventually, though, the polypeptide makes its way to the outer edge of the ER. And here it sort of pinches off in a tiny little membrane-enclosed sac, and it forms something called a transport vesicle. Now, that transport vesicle can take the polypeptide inside of it to a variety of places, but most of the time it takes that polypeptide directly to the Golgi apparatus. Once it's at the Golgi apparatus, the transport vesicle fuses and becomes part of the Golgi apparatus and releases the contents of the polypeptides that it's carrying into the Golgi apparatus. And then the polypeptide is traveling and moving through the Golgi apparatus, where more signaling sugars are put onto it, and where the Golgi apparatus may fold or bend or add components to that polypeptide. And eventually, That polypeptide makes its way to the end of the Golgi apparatus, and it gets pinched off in another little vesicle, but this is called a secretory vesicle because it's very secretory. Now, it's called a secretory vesicle because it's going to secrete something from the cell. It's going to secrete the polypeptide. That secretory vesicle moves away to the Golgi apparatus, away from the Golgi apparatus, and eventually meets the cell membrane and fuses with it. And when that happens, it releases whatever proteins it's made outside of the cell. That's the end of this video. Make sure you complete the corresponding review questions, and as always, be prepared to show me your notes in class. And here's your bunny of the day.